In this video, we're going to begin to talk in a little bit more detail about the steps that a virus has to go through in order to be replicated inside a host cell. So this is a diagram from your textbook, which is showing some generic steps of a viral replication cycle. So all viruses, now there's lots of diversity out there among viruses, and viruses use different kinds of host cells. They have different strategies to accomplish these different phases of their replication cycles. But this is just in a generic way providing an overview of the steps that all viruses have to accomplish. And a lot of this is just kind of common sense when you think about it. First of all, the viral particle is going to have to attach to a host cell receptor. So attachment is step number one. And again, virion is referring to a fully assembled virus particle that is capable of infecting a host cell. All right, and as we'll see, so a virus is going to have to attach to a host cell receptor. Host cell receptors are molecules at the cell surface on the cell membrane that the virus very specifically attaches to. So that's often the thing that dictates what the host cell will be. If a cell lacks the receptors that the virus attaches to, the virus is not going to be able to enter that host cell and replicate. So, and we'll talk a little bit more about that. The presence of the receptor is often what determines host cell specificity among viruses. It's not the only thing, but it is one of the key things. Then entry. So you've got to get the genome into the host cell because you need the host cell to read that genome and express the genes. And so there are different strategies that viruses can use to accomplish that, as we'll see. Sometimes the whole virion comes into the host cell. Sometimes just the genome, just the nucleic acids inside the core of the virus are released into the host cell. And so here we're seeing an example virus. It's an RNA virus, and its RNA is coming out of the interior or the core of the virus, being released into the host cell, and this RNA is undergoing replication because if you're going to reproduce the virus, obviously you got to make new copies of the genome. Every virus particle is going to have one of those. And over here they're showing you viral proteins being made. You got to make all the structural parts of your new viruses so that those can assemble together. So synthesis is the third phase. That's where you're replicating the virus's genome and producing all of these capsid proteins and glycoproteins that are going to be in the envelope. If we're talking about an enveloped virus, some viruses have matrix proteins in the middle. All of those things have to be made. Then you need to assemble new virions. So you got to package together new virus particles, and then finally those new virions or virus particles have to get out of the host cell. And the two key ways that can happen, some viruses do that through lysis, which means the host cell actually dies and bursts and the new viruses spill out. Other viruses go through a budding process where they push through the membrane, and a cell membrane wraps around the core of the virus and gives that virus an envelope. Remember, we said envelopes are phospholipid bilayers. They're cell membranes that the virus has stolen from its host while it's budding. All right, so again, let's talk a little bit more about these host cell receptors. When you guys take Biology 202, you learn about how hormones attach to specific receptors, and many types of hormones do that at the cell surface. Many hormones bind to a hormone receptor that's present on the surface of its target cell. And if that target cell doesn't have those receptors, then the hormone's not going to be able to go inside that cell and cause any kind of effect. And it's the same thing for viruses. They need some kind of receptor on the host cell surface that they're going to attach to. Very often those are proteins on the host cell surface or glycoproteins of some type. They might be carbohydrates of some type that are attached to the cell surface. All right, but viruses are generally pretty picky about their hosts. So, a virus that infects a dog, for example, usually is not going to be capable of infecting a human. Now, there are exceptions, like rabies virus, for example, can infect multiple species of mammals, but very often viruses are very, very specific about the hosts they infect. And not only that, but when they go inside their hosts, they're usually targeted to one particular part of the body. They can be very limited about the individual cells within the body that they're actually able to infect. And so that specificity is generally, again, determined by the receptor. And here's an example. So this is an HIV virus, human immunodeficiency virus, which causes AIDS. And on its surface, it's an enveloped virus. So in light blue here, this is an envelope. Here's the capsid. And on the inside, you've got two copies of the RNA genome that are packaged up inside there. But it has, protruding through the envelope, it has a glycoprotein called GP120. And GP120 attaches to a protein called CD4. And CD4 is present on helper T lymphocytes that we'll learn about when we cover the immune system later in the semester. Those are also sometimes called CD4 T lymphocytes because these CD4 protein molecules are present on the surfaces. Also present on macrophages, another type of cell involved in immunity in the body that we'll talk about later in the semester as well. So CD4 is key. If a cell does not have CD4 proteins on its surface, then HIV is not going to be able to attach to that host cell and enter. And later in the semester, we'll talk a little bit more about, or later actually in this chapter, we'll talk a little bit more about, on this diagram, you'll see these chemokine receptors here as well. And we'll go over in a little bit more detail about what that's involving when we're thinking about HIV. All right, so after a virus attaches to the host cell surface, the next key thing is to get the genome in there. And so there are different strategies for how that can happen. Here are a couple of examples. In this case, a virus is attaching to the receptor on the host cell surface, specific attachment, and then that whole virus gets engulfed. It's brought inside the cell, covered in a membrane vesicle, and then eventually that vesicle opens up, the virus breaks apart, and the genome is released. So this is saying free DNA here. Probably should say free DNA or RNA because it depends on our type of virus. Is that a DNA genome or an RNA genome that's inside the virus? And then another example over here, a different kind of virus attaches to the host cell surface, but instead of the whole virus coming inside in a membrane-covered vesicle, the envelope of the virus fuses with the cell membrane, and they kind of melt together, and that sends the capsid inside. The capsid breaks apart and releases the nucleic acid genome. So the genome's got to get out because we've got to have the host cell machinery see that genome and start going through the process of making the virus's protein so that we can assemble new viruses. Okay, so replication of viruses inside a host cell. So there are lots of different strategies by which genomes of viruses are reproduced or replicated inside host cells. And this is where we get into deviation from that story of DNA to RNA to protein to trait. Now, there are many viruses that follow that same pathway. Your DNA viruses that are double-stranded are going to do that. That's what they're showing you over here. Oops. If you have a virus that has a double-stranded DNA genome, when those genomes are released inside a host cell, there are genes on the genome, just like we have on our genome, or just like you'd find on a bacterial genome, and those genes have to undergo transcription. So you make messenger RNA from those genes, then those get translated to make protein. So that little green blob there is supposed to be viral protein, and there will be more than one kind of viral protein. So that's the same story we've already learned about in Chapter 8. And up here they're showing you, this is supposed to represent replication. That double-stranded DNA genome has to undergo replication, so you have more copies of the genome. So that's familiar. Now there are DNA viruses that are called positive-stranded DNA viruses. They're single-stranded. When that genome enters a host cell, the first thing that happens is you've got to make a complementary copy of that single-stranded DNA genome and convert it into double-stranded DNA. And once that happens, your host cell is going to treat that like a piece of its own DNA. Now that it's in that double-stranded form, it's recognizable. And so you'll have transcription of the virus's genes and translation to make the proteins of the virus. And then over here, we're seeing that more of these single-stranded plus strand DNA genomes need to be produced as well, because these are the ones that are going to get packaged up in the new viruses that are being assembled inside that host cell. So those are DNA viruses. Now on the RNA side, things are a little bit different. So if you guys remember, we have single-stranded RNA viruses, and those can fall into a couple of different categories. They can be positive strand RNA viruses, and they can be negative strand RNA viruses. And so we said that if a virus has a positive strand RNA genome, when that genome enters the host cell sees that genome as a messenger RNA. So a ribosome will attach and start translating the proteins of the virus. That's what they're showing you over here. You just go directly from the genome to beginning to make the proteins of the virus. But we also have to replicate this genome. Okay, so replication of nucleic acids, whether it's DNA or whether it's RNA, occurs through making complementary strands. You read a template and make a complementary copy by reading that template. So if you got a plus strand RNA genome and you need to make more of these, what you're going to have to do is there's going to be an RNA polymerase enzyme. RNA polymerases build RNA molecules that will first read this and make a complementary minus strand of RNA. And then it will read this to make more of these guys. I don't think they depicted that properly on the diagram here, but these are supposed to be more plus strand RNA genomes. So you're going to have, you start off with a plus strand RNA genome that enters the host cell. You make minus strand complementary copies. Then those are read by the virus's RNA polymerase enzyme to make more plus strand RNA genomes. And you've got a cycle that just keeps going on over and over here. Negative strand RNA viruses, the genomes are complementary to messenger RNA. So when those enter a host cell, you have to immediately make the complementary plus strand. You got to go from minus to pluses. And so that's supposed to be a plus strand RNA there. That can now be translated using ribosomes, using the host cell's ribosomes. Viruses don't have their own. And you make the virus's proteins. And then if you're going to make more of these minus strand RNA genomes, for replication purposes, you make plus strand complementary copies. And then the RNA polymerase will hop onto those and use the plus strands as a template to make new minus strand RNA genomes. Once you have those and you have the virus's proteins, you can assemble new viruses together. Okay, then you have double-stranded RNA viruses. So when those come in, you've got a plus strand and a minus strand already produced in complementary copies that are bound together, almost like you would have with double-stranded DNA. You need to separate those. You've got to have separated plus strands here that can be translated to make the proteins of the virus. And then you've got to undergo RNA replication to make more genomes to package up into new virus particles. Then your retroviruses, this is a separate category, and HIV falls into this category, and you'll hear about retroviruses. You'll hear that term used quite a bit clinically. Inside the virus particles, like we saw back over here, retroviruses have two copies of their RNA genome inside the virus particle. That gets released into the host cell. And retroviruses have a unique enzyme. It's called reverse transcriptase. And what that enzyme does, so if you think about transcription, what's transcription? You're going from DNA to RNA. So what would reverse transcription be? You're going from RNA to DNA. You're going backwards. That's why they're called retroviruses, because they violate that rule about going from DNA to RNA. They go from RNA to DNA when they first enter into a host cell. So they have a special enzyme that will hop on the RNA and read it, but synthesize a single-stranded DNA molecule, a DNA copy of RNA. And then that DNA single strand gets read to make the complementary DNA strand, and now you have a double-stranded DNA molecule. And then as we'll see later, once you have this, that is actually going to insert itself into the DNA of the host cell. It's going to integrate itself in there. And then the host cell just thinks this piece of retroviral DNA, of which HIV is an example, its genome becomes a double-stranded DNA molecule that gets inserted into the host cell's DNA, and the host cell just starts treating it like a set of its own genes. So this double-stranded DNA, you have transcription of the viral genes. There's a messenger RNA that gets translated to make the virus's proteins. And then also some of those plus-strand RNA genomes are going to wind up getting packaged inside the virus particle. So read through and take a look at that table and read in your textbook, read through the information there that describes the different strategies that these different classes of viruses use to have their proteins produced and to have their genomes copied, replicated, so that you can assemble new virus particles. Maybe now you're appreciating why we needed to cover genetics in Chapter 8 before we were able to do much of anything with this particular chapter. All right, so we kept harping on over here. You've got to make the virus's proteins. And again, viruses cannot make their own proteins. They don't have ribosomes and the other machinery needed to do translation. So that's one of the key reasons why they need a host. Viral genomes will have genes on their genomes, just like living things do. There's a lot of variation about how many of those genes exist. Those genes will code for structural proteins and replication proteins. So structural proteins are things like the capsid proteins or the glycoprotein spikes that stick out of the envelope if you have an enveloped virus, or matrix proteins. Some viruses have a matrix middle layer. So they're part of the actual structure of the virus itself. So the virus is going to have the genes that provide the blueprints for making those proteins, but the host cell is going to actually do the work of making those proteins. So these viruses are very clever. Then replication proteins, these are enzymes and other proteins that are needed for copying, replicating the virus's genome. Now some viruses will hijack the host cell's own machinery. If you're a DNA virus, some DNA viruses just use the host cell. You use your DNA replication machinery to make my genomes. Other DNA viruses have their own enzymes that are used to perform that function. With RNA viruses, think about this. RNA, we said over here, when RNA viruses replicate, they have RNA polymerase enzymes that convert minus strand RNA to plus strand and then back to minus strand. Or you go from plus strand to minus strand to plus, but you're always dealing with RNA there. So they've got RNA polymerases that read RNA templates and make new RNA strands. Our RNA polymerases and the RNA polymerases of other living things do transcription from DNA templates. They read DNA and make RNA complementary copies. So that's different from what these RNA viruses are doing. All right, so when we're thinking about, how are we going to combat viruses that enter the body? What kind of strategies do we need to use with our antiviral types of drugs. Fortunately, in many cases, viruses have enzymes that are needed for copying the genomes that are quite different from enzymes that our cells use to replicate our genomes or perform other functions like transcription. And we can take advantage of that and develop chemical drugs that will inhibit their replication enzymes while not screwing up our DNA replication enzymes or your RNA polymerases that do messenger RNA transcription. Now, in case you don't know already, we don't have, you think about it, if you get a virus, a lot of times they just kind of tell you we're going to treat the symptoms and you're going to have to ride it out. And that's because we don't have nearly as many drugs to treat viral infections as we do bacterial infections. And we're kind of lucky with bacteria because they're prokaryotes and we're eukaryotes. So there are enough differences between how their cells operate and our cells operate that we have quite an arsenal of chemical drugs that will mess them up without screwing up our cells. But when you come down, when you get to viruses, the options are a little bit more limited because they actually go inside our cells and hijack our cells for reproduction. So you really have to home in on specific enzymes of particular viruses and try to find chemical drugs that will inhibit only those enzymes while not harming the host cell. There are lots of things we could come up with that would shut down viral replication, but they would also kill you, and that probably wouldn't make a very good antiviral type of drug. Now, how about exiting? So we talked about how some viruses actually cause their host cells to burst open. That's called lysis. Many viruses do kill their host cells because they're hijacking all of this protein production machinery, and then the host cell can't make enough of its own proteins to survive. So many viruses do wind up killing their host cell either through causing lysis or simply by virtue of the takeover of the host cell machinery, eventually weakening and killing the host cell. But then some viruses get out through budding, which means they push themselves through a cell membrane in order to exit the cell. And so here we've got an example of that. So we've got, this is a virus with an RNA genome on this figure. These little purple things are supposed to be capsid proteins. So we've got an RNA genome that is in its protected capsid shell, and this looks like a virus with helical cell morphology. And then up here at the cell membrane, we've got these viral glycoprotein spikes that are already sticking in the surface, and these little blue dashes here are supposed to be viral matrix proteins. So those things are already embedded up in the cell membrane. The cell has manufactured those in a similar way to how it would manufacture its own membrane proteins, and it's sent those on up to the surface of the host cell. If you studied the endoplasmic reticulum and the Golgi apparatus in a previous class, that's how that all works. Proteins that wind up in cell membranes pass through the ER and the Golgi and then are sent on to the surface of the cell. So envelope viruses will hijack those processes as well and have their other proteins sitting up there waiting in the cell membrane. And then the capsid will attach to, the capsid is naturally attracted to these glycoprotein spikes or the matrix proteins if there's a matrix layer in the virus. And the capsid basically pushes itself through the cell membrane and those glycoprotein spikes, and that cell membrane piece wraps itself around the capsid, and that's called budding. Eventually that enveloped virus pinches off. So that's how an enveloped virus will acquire its envelope. That envelope is really it has stolen a section of the host cell's own membrane, and its little spikes were sitting up there already waiting, and now you have an envelope virus with its glycoprotein spikes sticking out. And so now we have an infectious virion with an envelope. That means this is a fully mature viral particle that can go off and find a new host cell to infect. This is an image that's was made with an electron microscope and colorized to provide more detail, but it's showing you here's a real host cell, and here are some real viruses that are pinching off from the surface as they're undergoing the budding process. All right, so that was kind of an overview of viral replication strategies. In the next video clip, we're going to talk about, because there's so much diversity among different types of viruses out there in terms of how they accomplish those different steps we just talked about, we're going to take a look at a few examples of specific viruses and their little quirky nuances for how they achieve those steps. So that'll be the topic for video lecture 4.